come to biology, the scientific study of life. Population conservation focuses on population size, genetic diversity, and critical habitat. Biologists focusing on conservation at the population and species levels follow two main approaches: the small approaches or population approaches and the declining population approaches. Small population approach, a species is designated as endangered when its populations are very small after such factors as habitat loss have taken their toll on population size. A population's smallness itself can drive it to extinction. Conservation biologists who adapt the small population approach study the process that can cause very small populations finally to become extinct. The extinction vortex. A small population is prone to positive feedback loops of inbreeding and genetic drift that draw the population down an extinction vortex toward the smaller and smaller population size until no individuals exist, as shown this figure. The key factors driving the extinction vortex is the loss of the energetic variation necessary to enable evolutionary responses to environmental change, such as new strains of pathogens. Both inbreeding and genetic drift can cause a loss of genetic variation, and the effects of both processes become more significant as a population shrinks. Not all populations are doomed by low genetic diversity. Low genetic variability does not necessarily lead to a permanently small population. For example, overhunting of northern elephant seals in the 1890s reduced the species to only 20 individuals, clearly a bottleneck with reduced genetic variation. Since that time, however, the northern elephant seal populations have rebounded to about 150,000 individuals today, though their genetic variation remains relatively low. Furthermore, a number of plant species such as the lawswort, pedicularis, and several grasses seem to have inherently low genetic variability. Many populations of cordgrass, Spartina angelica, which thrive in salt marshes, are genetically uniform at many loci. S. angelica arose from a few parent plants only about a century ago, and hybridization and alubulai having spread by cloning. This species now dominates large areas of tidal mudflats in Europe and Asia. Thus, in some cases, low genetic diversity is associated with population expansion rather than decline. But these cases may stand out preciously because they are so unusual. How small does a population have to be before it starts down an extinction vortex? The answer depends on the type of organism and several other factors and must be determined case by case. For example, large predators that feed high on the food chain usually require very large individual ranges, resulting in very low population densities. Those not all rare species concern conservation biologists, whatever the number. However, most populations presumably require more minimum size to remain variable. Case study the greater prairie chicken and the extinction vortex. When Europeans arrived in North America, the greater prairie chicken, Tympanucho copios, was coming from New England to Virginia and all across the western prairies of the United States and Canada. Agriculture later fragmented the populations of the greater prairie chicken in the central and western states and provinces. For examples, the Illinois alone greater prairie chickens numbered in the millions in the 19th century, but declined to 25,000 birds by 1933. By the 1993, the Illinois population of prairie chickens was down to only 50. Thought large populations remained in Kansas, Minnesota, and Nebraska. Researchers found that the decline in the Illinois population was associated with a decrease in fertility. As a test of the extinction vortex hypothesis, the scientists imported genetic variation by transplanting more than 270 pairs from larger populations in Kansas, Minnesota, and Nebraska. The Illinois population rebounded, confirming that it had been on its way down the extinction vortex until rescued by a transfusion of genetic variation. Minimum variable population size. The minimal population size at which a species is able to sustain its numbers and survive is known as minimum variable population, MVP. MVP is usually estimated for a given species using computer model that integrates many factors. The calculation may include, for example, an estimate of how many individuals in a small population are likely to be killed by small or by some natural catastrophe, such as a storm once in the extinction vortex 2 or 3 years in a row of bad weather could finish of a population that is already below MVP. The MVP of population is factored into what is called a population variability analysis, PVA. The objective of the analysis is to reasonably predict a population's chances for survival, usually expressed as a specific probability of survival. For example, a 99% chance over a particular time, for instance 100 years. Modeling approaches such as a PVA allow conservation biologists to explore the potential consequences of alternative management plans because modeling depends on reliable information about the populations under study. However, conservation biology is most robust when theoretical modeling is combined with field studies of the managed populations. The case study on grizzly bears is an example of such an approach. Um, Effective population size. Genetic variation is the key issue in the small population approach. The total size of a population may be misleading because only certain members of the population breed successfully and pass their alleles on to offspring. Therefore, a meaningful estimate of MVP requires the researchers to determine the effective population size, which is based on the breeding potential of the population. The following formula incorporates the sex ratio of breeding individuals into the estimate of effective population size, abbreviated by NE. Notice that NE equal 4 NF, NM, distribution NF plus NM. So F, which means female, M masculine or male, where NF and NM are respectively the number of females and number of males that successfully breed. Applying this formula to an idealized population whose total size in 1,000 individuals, NE will also be 1,000 if every individual breeds, and the sex ratio is 500 females to 500 males. In this case, NE equal 4.500 distribution 500 plus 500 equal 1000. Deviation from these conditions reduces NE. For instance, if the total population size is 1000 but only 400 females and 400 males breed, then NE equal 4.400 distribution 400 plus 400 equal 800 or 80% of the total population size. Numerous life history traits can influence NE, and alternative formulas for estimating NE take into account family size, maturation age, genetic relatedness among population members, the effects of gene follow between geographical separated populations and population fluctuations. In actual study populations, NE is always some fraction of the total population, thus simply determining the total numbers of individuals in a small population does not provide a good measure of whether the population is large enough to avoid extinction. Um, Whenever possible, conservation organisms are geared to sustain total population sizes that include at least the minimum valuable number of reproductively active individuals. Um, the conservation goal of sustaining effective population size in a above MVP stems from the concern that populations retain enough genetic diversity to be evolutionary adaptable. Case study analysis of grizzly bear populations. One of the first population variability analysis was concluded in 1978 by Mark Schoffer of Duke University as part of a long-term study of grizzly bear in Yellowstone National Park and surrounding areas. A threatened species in the United States, the grizzly bear Ursus rectus horribilis this is the scientific name. Is currently found in only 4 of the 48 contiguous states. Its population in those states have been drastically reduced and fragmented in 18 and estimated 100,000 grizzlies ranged over about 500 million hectares of more or less continuous habitat. While today there are 6 relatively isolated populations totaling about 1,000 individuals with a total range of less than 5 million hectares. Schoffer attempted to determine variable sizes of the U.S. grizzly populations using life history data obtained for individual Yellowstone bear over a 12-year period. He stimulated the effects of environment factors on survival and reproduction. His models predicted that given a suitable habitat, a total grizzly bear population of 70 to 90 individuals would have about a 95% chance of surviving for 100 years, whereas a population of 100 bear would have a 95% chance of surviving for 200 years. How does the actual size of the Yellowstone grizzly populations compare with Schoffer's estimates of a minimum variable population size? Several sources of information indicate that the grizzly population of Yellowstone is growing. On the best indicators of that growth is the number of females observed with a calf each year. Calves each year increases in this number from 1973 to 22 support that conclusion.